Hello everybody, I'm Peter Cooper from the NCBI Service Desk. Welcome to today's webinar on NCBI resources. The materials are available for this webinar, including the slides, the demonstrations I'm going to do, and the script for this. Not to say that I'm actually following that script. They're all available at that compressed URL on our FTP site, and I'm going to send that in the chat pod to everyone that's listening, so you should get that. The other thing that you're going to see in a lot of my slides, in case it's not obvious, I've abbreviated some of the base URLs for some of these links. So if you have the NCBI in angle brackets, that means the URL of the NCBI homepage goes in that position. If you see the FTP in the angle brackets, that means that the FTP site's address goes there. And of course, YouTube's address goes where you see the YouTube in angle brackets. Okay, so today's topic is the newly released multiple sequence alignment viewer. I'm going to call it MSAV mostly during this talk just to make it easier to say. This is available on the NCBI alignment services, and as we'll see, it can also accept your own multiple sequence or alignment data. You can upload it into this viewer. So we'll talk a little bit about what the multiple sequence alignment viewer is, the purpose and scope, what its features are, how to access it from NCBI tools, how to upload your own data, and then I'll do some live demonstrations on how to do things. Okay, the multiple sequence alignment viewer is a general purpose alignment viewer. And as my colleague Tao is reminding me, just to point out to you, this is just a viewer, so it doesn't perform a multiple sequence alignment for you. We'll look at a tool today called COBALT that will do that. It's useful for assessing sequence conservation, variation, or errors in protein and nucleotide alignments. It has lots of useful coloring schemes that can emphasize different aspects of sequence conservation or divergence. The viewer plugs into NCBI alignment tools, but also allows you to upload your own data. And as with the related sort of NCBI viewer called the sequence viewer, the JavaScript for the MSAV is available, and you can embed it into your own web page displays. And I'll show you where that link is a little bit later on. I'm going to go over some of the MSAV options that you can use on the display. So one of the very useful things to be able to do if you're working with NCBI sequences is you can expand any row in the viewer by clicking on the plus sign there, and the expanded display will show you annotated sequence features, conserved domains, binding sites, name sequence motifs, and so on. And we'll see in the demonstration in a few minutes. This is very useful for mapping features onto annotated or poorly annotated sequences in the alignment. Of course, in order to see these features, as I already said, you've got to be working with NCBI accession sequences, and we will be doing that today. The viewer does work with other kinds of multiple sequence alignments, but it won't be able to retrieve the information from NCBI unless you use our accessions. Another feature that's very useful is being able to color your alignment. This shows the coloring schemes that are available for protein alignments. You can access the coloring options and other controls through the Tools menu up at the upper right, or you can right-click anywhere on the alignment display. The coloring schemes are going to emphasize aspects of a column in the alignment, such as amino acid properties, conservation, or variation. Many of these can be set to show departure from the column average or from a designated master sequence. The nucleotide alignment coloring schemes are a little bit more simpler than these are, and we'll take a look at both of these in the live demonstration in a few minutes. Another feature is being able to click on a particular residue and to get some context about where it fits into the alignment and where it fits into that particular sequence. So you can just use the mouse pointer. If you point over a particular residue, that pop-up will appear. The information includes the position in the alignment, the position in the sequence itself, statistics on the column, such as the number of matches, mismatches, and gaps. And the pop-up also provides overall statistics for the sequence in the alignment, such as all the positions that are included in the alignment. Okay, so how do you access the MSA viewer? You can access MSAV from alignments generated at NCBI from BLAST. From COBALT, it's also available now from the virus variation pages, from pop sets that have submitted alignments, from BLAST results, and that says coming soon, but that feature was released yesterday, so we will see that live today. You can also use the projects page for the MSA viewer, and the URL for that is shown at the bottom of this slide. And you can also specify things with URL parameters, and some of that's demonstrated on the project page itself. So let me just highlight a couple of these points of access before we go into the demo. So we talked about generating multiple sequence alignments a minute ago. One of the tools that we have for doing that is COBALT. COBALT generates a true multiple sequence alignment. You can launch COBALT from its submission page. The URL is shown here on this slide. Or you can launch it directly from a set of BLAST results. BLAST results are interesting because they let you collect your related sequences first and then perform a multiple sequence alignment with them. And so what you get on the COBALT results now is sort of as a graphical viewer embedded into the page is a multiple sequence alignment viewer. And it gives you an interactive display that lets you do the kinds of things we talked about before within the COBALT page. The other place that you can get to the MSA Viewer directly from results at NCBI is from the BLAST results now. And this is a set of nucleotide BLAST results, so you'll see there's a link there under Other Reports that goes to the MSA Viewer. And when I click on that, it opens up the MSA Viewer, which is really an alternative viewer for BLAST results. It's much more interactive than the BLAST graphical viewer that's there now. So I talked about using URL parameters. Notice that you could simply pass the RID directly to the MSA Viewer application to see your BLAST results in this way. Speaking of that kind of thing, this is the MSAV project page, and the URL for it is listed on the top here. The project page provides access to the application itself through the upload button, and we'll use that to upload some data today. Projects page also has some examples, so you can click on those links at the top just to see what it does with various kinds of data. And then I talked a little bit about embedding the MSA Viewer into pages. There are some demos of what you can do with that at the bottom of this page, and the MSAV API page has complete instructions on how to embed this in your own web pages. Okay, so this is what the upload interface is like. Notice it's very similar to, it's essentially identical to the one that's available for our various genome browsers, plus the graphical sequence viewer. You can upload a BLAST or COBALT RID by simply entering that if you click on the BLAST results. You can upload an outside multiple sequence alignment through a data file, a URL, pasted text even. The formats that it accepts are FASTA plus gaps, and that's the default format that the multiple sequence alignment program Muscle produces, and that's a very popular program. There's the URL drive5.com/muscle if you want to download that and work with it. It also will accept ClustalW format, another sequence alignment tool that's very popular, and a seq-align, which is the NCBI ASN1, which is generated by our tools like COBALT. Before I go on to the demonstration, I just want to remind you that the MSAV is under development. And so there is a feedback link here that lets you provide suggestions or bug reports, those kinds of things. So please do so if you have any ideas about how we could improve this viewer. I also want to remind you that you can write to us directly too. My email address is there, peter.cooper@nih.gov. For questions about BLAST help or problems with BLAST help, write to BLAST. Write to blast-help@ncbi.nlm.nih.gov. For general help, write to info@ncbi, etc. And then if you have questions about this webinar in particular, technical aspects of it, when's the recording going to be available, those kinds of things, you can send that to the webinars address. Okay, so we're going to do the tricky part now. I'm going to leave the slideshow and do some live examples. The examples that I'm going to follow are included in that materials directory that I sent to everybody. The steps are there in case you want to follow along with me or if you want to try this at a later time. I'll use the protein and nucleotide alignments to demonstrate the features of the MSAV. For the protein alignment, I'm going to start with a set of related serotonin receptor sequences from the NCBI protein database that I collected ahead of time. I'm going to align them with COBALT and just use the embedded viewer to demonstrate some things. For the nucleotide sequences, I'm going to start with a BLAST result for mammalian albumin mRNA sequences. I'm going to load those into the viewer. Then I'm going to take the same sequences and that I've made a multiple sequence alignment with those, and I'm going to upload that into the viewer to talk about some differences between those two things. Okay, so I'm going to leave the slideshow and switch over to my web browser right now. So the first example is a set of 12 proteins. I got these proteins by looking at orthologs from the annotation pipeline, and I honed it down a little bit, got rid of gene model sequences just to make a simpler set. These are all serotonin receptors, the to a protein product from tetrapods, from a group of mammals, and I think there's one chicken sequence in here. So what I'm going to do is I'm going to align them with COBALT. So this is a way of just throwing something directly into COBALT and aligning it. So now all my accession numbers are in here. Of course, you can put sequences in here as well. And I'm going to align that. And we're going to look at the viewer as it's embedded on the COBALT results. And COBALT doesn't take very long to align a set of sequences like that. So you can see that the MSA viewer is embedded at the top of the viewer here. The rest of the COBALT results are here. I'm not going to spend much time talking about those today, but it has descriptions and it has sort of a text alignment at the bottom. We're going to focus on the viewer, of course, which is embedded at the top here. I've got the accession numbers on the right, the species names, that's the left, my left, and the species names are on the right. And then I can look at the sequence. Let's zoom into a particular region. This is the default coloring scheme, which is by conservation. It's a fairly strict sort of a black or white, or really blue or red kind of a thing. I'm going to zoom into this region over here where we can see a little bit of blue. So what I'm going to do to zoom in is I'm going to click on the sequence here, viewer itself, and I'm going to zoom to sequence. And so you can see that the rest of these columns are considered conserved, although you can see there is some variation in the amino acids that are there. And then this is considered kind of a mixed bag here of different kinds of residues. There are lots of ways to color this. I can access the coloring from the Tools menu. I can also simply right-click on the background here and change the coloring. So I can get things about amino acid properties. For example, rasmol amino acid colors are kind of interesting and colorful. This is more about the kind of amino acid it is. Other kinds of things that I can do are to show aspects of conservation in terms of blossom scores or in terms of differences. So I could do a frequency-based difference like this, which is basically comparing this to the rest of the column. So the redder the residue is, the more different it is from the rest of the column. And then the other thing I can do, for example, is I can do difference from a master sequence. Let me just show you what happens if I do that without setting a master sequence. Show differences. Notice that I don't see any differences. So let's go ahead and set a master. So the second sequence is the human sequence. To set a master, I simply right-click on the sequence and set the master. And now you can see where there are differences. These are all compared to the human sequence, and I'm just showing where there are differences from the human sequence. Let's go ahead and expand the first row here, which will give me the features from that human sequence. And I can see there's a feature here, but what I'll do is zoom out so we can see a lot of different features that are present here. So I'm just using the zoom graphic to do this. This is a G protein coupled receptor, also called a 7-transmembrane receptor. And you can see that that conserved domain 7TM_1 is present there. There are also the transmembrane regions are marked. There are 7 of them. They're there at the bottom. And then there are other kinds of active sites or binding sites. The agonist binding site is highlighted for you, as well as some motifs that are present in these sequences. So you can play around with coloring some more if you want to. Let me just show you one more coloring thing that's kind of interesting. In this particular case, we would expect the transmembrane domains to be hydropathic. So we can have this hydropathy scale here. And what that highlights for you, the redder the residue is, the more hydropathic it is. And you can see that they do a pretty nice job of highlighting the transmembrane domains where those red areas are. The other thing that's kind of useful to be able to do here is we have a very well annotated sequence at the top. The sequence right below it, the chicken sequence, if I expand the information on the chicken sequence, it does have those large conserved domains mapped onto it or annotated on it, but it doesn't have the other parts. So I could use the human sequence as a model to simply map these kinds of features onto that chicken sequence. So for example, let's zoom into this NPXXY motif here. Let me just go ahead and zoom in there. And I'll go ahead and change this back to show difference. In fact, there are no differences from the human sequence in this region. And so suppose I want to know what residue in the chicken sequence corresponds to that motif. So if I want to sort of figure out exactly which asparagine it is, I put my mouse pointer over that residue, and I'm just going to put that pin down so it stays there. So notice that this is a asparagine that's at position 373 of that sequence. So I could use this as a way of mapping directly the particular feature onto the sequence. Right, so let's shift gears a little bit, and now I'm going to upload an alignment file. But first I'm going to show you the BLAST results that I used to collect those sequences. So to do that, I'm just going to show you a BLAST result that I have saved by an RID. And this RID is in your FTP site handout, so you can just go to recent results and retrieve it. The URL to retrieve it is also there. These will be saved for a while. And so you'll see that I have the ability to load this directly in the MSA Viewer right here. You go ahead and open that. And so this is a set of BLAST alignments that are opened in the MSA Viewer. So this is basically just a search of the RefSeq database on BLAST using the human albumin mRNA as a query and limiting it so that I get rid of model sequences. So I just get the NM style reference sequences. So the coloring options here for a nucleotide alignment are similar to the ones we saw before, but they're more limited. So the one that's here is a very useful one for this. It's frequency-based differences. It just shows you how a particular column, how uniform it is in terms of the residues that are there. And of course I can do show differences if I want to as well once I set a master sequence. So let's go ahead and take a look at a particular part of this alignment. I'm going to go in here at position about 550. So I'm going to zoom to sequence right there. So one of the things to point out here that this is a BLAST alignment. I'm going to show you an example of where we can see that that's the case. So remember, this is really just a stacked pairwise. Everything is aligned only to the query sequence. They're not aligned to each other. There's no optimization of the distance between all the sequences. And so this is a little place where you can actually see that, because this can actually result in apparent misalignment of closely related sequences. And I can see that right here if I look at position 562. Notice that there's a TAA here in the cow sequence. In the closely related sheep, there's also the same set of sequences, but there's a gap inserted here. And that's because these two sequences haven't been optimized. If I did a multiple sequence alignment with this, these would be aligned better to each other. In fact, that's the way multiple sequence alignments programs work, is they cluster the sequences by how similar they are to each other and then compare those groups to the other groups. Okay, so the last thing I want to do is to upload a multiple sequence alignment. So what I'm going to do is to just chew back the URL here all the way back to the MSA Viewer projects. And notice that that projects page really is, if you put a question mark on the end, you're basically accessing the application. So this is the projects page that I showed you in the slideshow a few minutes ago. So I'm just going to click the upload interface here. And now what I'm going to load is a multiple sequence alignment of these sequences. So I'm going to pick a data file. Notice I could also, if I had them on a web page, I could load the URL. If I had them as text, I could paste them directly in here. I'll just do the data file option here. And I have those available to me on this hard drive. So this is simply a FASTA plus GAPS alignment that I made using Muscle. So now I've uploaded a multiple sequence alignment, and you can see right away, you'll notice that one of the differences is that all the residues are included in this alignment. One of the things that I didn't get to, but you can try it yourself, if you look at the mouse sequence in that last alignment, it wouldn't have all been aligned. Only part of it was, because that was a local alignment. This is a global multiple sequence alignment. Let's look at position 540 in this alignment. And what I can see is we can look at a couple of things here. You can notice that there's a group of sequences here that has a gap inserted there. Those are all from a group of mammals that are in the Laurasia theria, so those are more closely related to each other. And you'll notice that that's how these are done. So that's because this is a multiple sequence alignment. It grouped related sequences together first before it made a global alignment. So we have rodents first. We have the pig, the sheep, the cow, a couple of horse-related things together as Laurasiatheria. And then we have some primates and some carnivores there. So we can look at this to see that this is indeed now a multiple sequence alignment. If we look at the mouse sequence, which I didn't show you in the previous one, but you can find it up here. If I put my mouse pointer over that, notice that the sequence is entirely aligned from the first base to the last base. On the previous one with BLAST, it was only partial alignment because the ends of the sequence didn't align very well to the human sequence. Multiple sequence alignments means that it will align anything to anything, even things that are unrelated to each other, so keep that in mind. One of the things I'll just show you is a display option that's a little bit confusing at first until you explain it a little bit. This business of having a master sequence is important, but one of the things that the master sequence does is it's forced to not have any gaps in it. So I'm just going to promote the cow sequence to the master position. And you'll now notice that those residues from all the other mammals that occupy the position of that gap in the cow sequence are now essentially gone. They've been deleted. And they're represented by that blue highlighting with the blue vertical bar there. And notice that the other members of the Laurasiatheria then are not gapped there. So that is basically what I wanted to show you today with the multiple sequence alignment viewer. And I'll return briefly to the slides and we may have time for a couple of questions. So let me just finish up with a slide here that gives you some useful links. And some of them we've already mentioned previously, things about the MSA Viewer, how to embed the MSA Viewer. Remind you that to get the most recent recordings of our webinars, please visit our YouTube channel. And there is an introductory YouTube video on the MSA Viewer. We have a lot of helpful fact sheets, and you can go to our Learn page to get access to those as well. Okay, so I'll stop here and see if we have any questions. I'll leave it open for about 5 minutes and just see if anybody has anything. Okay, so the question, and that's a good question, whether you could mix FASTA sequences with accession numbers in COBALT, and apparently that does not work. Okay, and somebody asked me to repeat the question again. The question was whether you could put a mix of, in COBALT, whether you could put a mix of accession numbers, bare accession numbers, and FASTA formatted sequences, and the answer is no, you cannot do that. Okay, so that concludes the webinar for today. Thanks everybody for listening, and we'll see you next time. I'll send out an email to everyone who registered when the recording is available.